Okay, so in this video, we will be covering some key concepts that are related to DNA, RNA, gene expression. We're also going to cover a couple of the Shafgar rules, gene expression, mutation, and going to name a couple of the most important historical figures with DNA. Okay, so let's go ahead and start with share screen here. So we have these Shafgar rules. So the Shafgar rules, these are rules that are for DNA. Additionally, these say that there should be an equal quantity between these bases. So for the DNA, there are 4 different bases. We have adenine, thymine, guanine, and cytosine. For each of these bases, there's 2 that pair up with each other. We have adenine and thymine, so they're both together, and then we have guanine and cytosine. A little memory trick for this is that apples are in trees and cars in garages. So adenine will always pair with thymine, and cars or cytosine will always pair with guanine, okay? Another thing is that, here's an example that thymine, if it makes up that 15% for that particular DNA sample, adenine would also be that 15%, okay? So don't forget that. Righty, so the next thing, let's briefly discuss some experiments and discoveries. So we have the Hershey and Chase experiment. So this experiment showed that bacteriophages, so these are viruses that are infected in bacteria, they inject only their DNA, so not their full protein, but their DNA, only for that particular host. So this is something that was very pivotal in discovering and proving that DNA carries that genetic information. Then we have Rosalind Franklin. So she used something called X-ray crystallography. So she used that to identify the physical characteristics of DNA. So her work was fundamental in discovering the shape of DNA. So if we go back and think about what we learned in class, so she was that person that ended up working really, really hard and didn't mind sharing her information with Watson and Crick or other scientists. She ended up dying of ovarian cancer because she used all that radiation. Then we have Watson and Crick. So these 2 people are credited with discovering the double helix structure. So they were the first people that created that model of what that DNA looks like, okay? So a good representation of that genetic material. Okay. So now let's compare DNA and RNA. So DNA is a double strand. So I want you to think of that D for double strand. So it has 2. So D for double strand and RNA for single. Okay, so it's just 1. So DNA has that double strand. It contains that base thymine. So going back to our little tree here. So we have apples going to trees, cars, and garages. So they have, and DNA has that thymine. RNA, instead of thymine, uses uracil. And instead of deoxyribose sugar, they only have ribose sugar, okay? So think of that R for RNA ribose sugar, okay? Right. Now, moving on to human keratopes. So humans, we have 46 chromosomes in total. So these are organized into 22 pairs of autosomal chromosomes and then 1 pair of sex chromosomes. So for females, it'll be XX. For males, it'll be XY. Make sure you know a karyotype. So a karyotype is an image that shows pretty much an individual set of chromosomes. This is used to detect abnormalities or study specific genetic traits, okay? Chromosome structure. So here's a breakdown of the chromosome structure. So DNA, it wraps around the proteins. These are called histones. So here are the little guys over here. They're little circles. So they are used to form what we know, the chromosomes, okay? Autosomes, so autosomes here are chromosomes that are not sex chromosomes. And a diploid, so we studied this a little bit in class, making sure you know that root word of that prefix. So diploid, di means 2. So diploid contains 2 sets of chromosomes, and then it's 1 from each parent, okay? Now, DNA replication and mutation. DNA replication is a semi-conservative process. So this means that each new DNA strand contains 1 original strand. So on 1 strand. So this means that it's pretty much just copying that template and pasting it over, okay? So this is a really critical process. And now primer. So we talked about this in class. So think about whenever you're painting, you need a primer so your paint can stick. The same thing as here. So we have a primer, which is a short piece of nucleic acid, and it attaches to DNA to initiate that DNA replication. So you need that little part, that little primer, in order to stick and create that whole DNA replication, okay? Now we have mutations. Make sure you know that mutations are a permanent change. This is a permanent change in the DNA sequence of a chromosome, okay? And now base pair substitution. So this can occur, so this can change pretty much the RNA sequence. So we have the original RNA would be CUU, GAU. The mutated, we can change that A to a U, so it would be a whole different RNA strand. Insertion, think of it's inserted or it's inside, so 1 or more nucleotides are inserted into that DNA. Deletion, think of Control-Alt-Delete, or it's deleting. So thalassemia, this would be an example of a disorder that's caused by deletion of mutations that pretty much affect the hemoglobin genes. Okay. Now we have gene expression and translation. So the first thing is that we have transcription. So DNA is transcribed into mRNA. So DNA or transcription has an mRNA that helps with RNA polymerase. Translation has that 3 base sequences. So mRNA, it's translated into these proteins. So in translation, we have these codons, which are the 3 base sequences. And then they specify which amino acids can be used to make that protein. So transcription, think of you have like a paper and you're transcribing that information. Translation, you're getting that paper and you're translating what that means, okay, and what that's specifically going to be a result of. Codons and anticodons. So codons, they have that mRNA. So mRNA, codons have mRNA, so the three mRNA bases codons for specific amino acids. Anticodon, think of that T in anticodon, is a sequence of, again, 3 bases on tRNA. So the tRNA, anticodon, so tRNA that pairs with that codon and mRNA. So this ensures that the correct sequence of amino acids in a protein are being produced, okay? Introns and exons. So we need to understand the differences between introns and exons. Introns are non-coding regions of a gene. They're removed before translation. So think of interruption. So introns are interruptions. Exons are codings of regions of genes that are actually expressed in the final protein. So think of exons, they exit the nucleus to be translated into proteins. So introns, think about, you have your things that are in a little meeting and then they interrupt to let them know that something needs to be removed. Exons, they wait until the final process to exit or exit so they can be expressed, okay? So introns interrupt while exons, they express. And then important concepts to understand. So DNA to RNA, this would be a transcription example. So we have 3' GAT, CC, ACT, 5'. So the corresponding RNA, so remember this specifically for the test. So RNA does not carry that T, it carries that U. And you have to make sure that your strand is going to be flipped. So that 5' is going to be beginning. So you're going to be pairing them backwards, okay? And then remember the uracil. So instead of that thymine for that DNA, RNA has that uracil. So you're pairing those two, okay? And that is it for today. If you have any questions or need additional support, let me know.